Mercury is just a challenging and it's a fascinating element, but it's a very challenging one in that there are various forms of mercury. You have elemental mercury, which is what most people think about when they think about mercury. It's that silver kind of colored sort of bead look that's used to be in thermometers. And that mercury has various physical characteristics that makes it very challenging. It's very dense material. So if you have mercury in soil and you dig a hole, you need to try to remediate the soil or have any other kind of impact to the soil environment. That elemental mercury will go to the bottom of the hole, and then it is susceptible to getting into groundwater and shallow subsurface flows. If you have mercury in a building, it inevitably will end up in the basements, and some of the old basements that connect very closely with the groundwater surrounding the building. That can be a challenge. And mercury in storm drains always finds cracks and crannies into the old storm drain pipes and finds its way into shallow subsurface. So its physical characteristics are challenged. Mercury is easily methylated by microbes in the environment. So again, that's where you have these complexities of the chemical and the biological. Once those microbes methylate mercury, that creates a more toxic form called methylmercury. And that is easily bioaccumulated, so it moves up into the food chain. So it moves from microbes, say, to algae, to invertebrates, to fish, and eventually can be a toxicological concern to humans and wildlife.
Knowing the composition of the types of species that are in the community is really important. And this long-term biological monitoring program has informed our mercury technology development project in many ways. First, the types of species accumulate mercury very differently. And so they actually could influence the mercury cycle in different ways. And that when we have mercury that flows from the algae communities up through the food web, the types of species that are in the community can influence how much mercury is transferred up the food chain. So knowing who's there is really important.
We sample the same sites over time in a mercury contaminated stream. And because of this, we are looking at trends in mercury bioaccumulation over time and how fish respond to changes, different remediation actions that are happening that remove mercury from the water. We have, over time, seen the same fish time and time again 4 or 5 years later, and then we're able to take another sample and see not only how has that fish grown, but how much mercury has accumulated in that fish over time. So we're getting real-time results on bioaccumulation rates in the field.
One of the things that we're trying to look at here in the Aquatic Ecology Laboratory is to try to develop ways that we can not only reduce the amount of mercury going into the system, but also can we modify the stream environment? Can we modify the chemistry and the biology to maybe reduce that uptake of mercury into the food chain?
Freshwater mussels are filter feeders, so they feed by pumping large quantities of water over their gills, and they're looking for algae and other particles that they can feed on. And we actually are looking at the potential for mussels to filter not only the particles, but the mercury that's in the water. So mussels, because they filter such a large quantity of water, they actually can remove mercury from the water column. And we're looking at this to potentially take mercury out of the water column so that it's not then available for bioaccumulation into fish.
From water chemistry to biological, to the use of sorbents and impacting erosion that goes into the creek. All of those are sort of technology type solutions that we're evaluating here in the Aquatics Lab, using the stream mesocosms here behind me to try to develop solutions for environmental management.